All right, welcome to part 2. So I'm going to actually kind of be stepping through a lot of the materials that are within this lab. There will be an opportunity to turn in a couple pages at the end. But the second question on the pop bead asks, is it possible for a restriction site to be within a gene, and would the gene still be functional? This kind of goes back to that description I just had a couple slides ago, where you can absolutely have those recognition sites within a gene. And so in this case, you've got your green restriction site here. And of course, it's right in the middle of the LAC-Z gene. But the LAC-Z gene will still be functional unless you've cut with the green restriction enzyme. So just kind of keep that in mind, is that it is possible, and it's likely, for restriction sites to be within a gene, and unless you've actually digested them with that restriction enzyme, it should have no effect on the functionality of that gene. Now the next part of the lab asks you to orient the plasmid as it's shown in the map, and then to think about your hands being the restriction enzyme. In this case, we're going to be talking about the restriction enzyme for yellow. What would that enzyme do if it interacted with the plasmid? And so have the yellow cut the DNA. And so now you should have, and by cut, I mean you're just going to open up your plasmid so that you've got a yellow bead laying on either side as you lay it out in a linear fashion. And this is not unlike what happens when you cut a plasmid with a restriction enzyme. It linearizes the plasmid so that now instead of it being in a circle, it's actually in a linear form. And so now, you want to think about how to draw the map of this resulting DNA fragment. And so even though you have cut with this yellow restriction enzyme, you do want to show that there is, that partial restriction site at either end as you go about drawing your map. So as you go about drawing your map, it should look something sort of like this, or something exactly like this. If this is your bead set down here, this is your ampicillin gene. Remember, the blue is your LAC-Z. You've got your yellow kind of designated at the very end, and you see all these things actually on the plasmid map that's been drawn. So you've got your yellow restriction sites at either end of your linearized plasmid. You still have your green restriction site there in the middle of your LAC-Z gene. You've got a pink restriction site somewhere in between the LAC-Z and the ampicillin. You can kind of, I think, see how these two things relate. How you've got your beads that are kind of mirroring what that restriction map or what the actual plasmid map that's been linearized should look like. And hopefully this makes sense to you. When you actually draw this out, it's also very helpful to be writing in the different sizes of the fragments in between those restriction sites, just because it can help you map later, make sure that you are keeping track of everything appropriately. Because that's where sometimes it's kind of like a puzzle, and it can get a little confusing if you're not really keeping track of stuff well. So now it says to go ahead and recircularize your plasmid, and question 4 asks, what is the size of your plasmid? And so this is nothing super complicated. You've been given some restriction information here, how big the pieces are between pink and green. This is 750 base pairs. Between green and yellow is 850 base pairs. And then from yellow all the way back over to pink is another 1400 base pairs. This is not complicated. In order to determine what the size of your plasmid is, You're just going to add up those numbers. You're going to take them and you're going to sum them, and that's going to give you the overall size of your plasmid. So when you do that, you end up with 3,000 base pairs is the overall size of your plasmid, and that's usually added in the middle portion of your map. It's always very useful to have the size of the plasmid, and then as you do subcloning steps or other manipulations, you can always kind of know where you started and go from there. Some of the other questions then, question 5 is asking you about how many genes are there on the plasmid, what are their names, how many restriction sites. Again, this is not super complex, but working through this a little bit at a time, it seems very simple. It's when you try to tackle it as one big entity that it becomes a little overwhelming. And so when you kind of break it down into the pieces, I think it makes it much more digestible, if you will. And so the genes we've talked about. We've got a LAC-Z gene that's on the plasmid here that will be functional and will produce a blue colony for any bacteria that take up this plasmid. And then we have another gene that's an ampicillin resistance gene, which enables any bacteria with this plasmid to grow on an ampicillin media. As far as the restriction sites, there are 3. We've got a yellow restriction site, a green restriction site, and a pink restriction site. And this would be very similar, in an actual plasmid. Instead of pink, green, and yellow, it would be things like EcoR1 or NOT1 or XHO1. You're going to have an actual enzyme name and not just a color. The colored beads help simplify everything though. Now, the protocol is asking you to cut with the pink enzyme and then to draw a map. So again, on your plasmid, you're going to open it up between the 2 pink beads and lay it down so that the whole end is on the left. And then you're going to be able to draw a map based on what you see in front of you with the beads. And that map should look something very much like this, where you've got your pink. Again, you're making sure that you're including that pink restriction site at both ends of the linearized plasmid. You've got your LAC-Z gene, which is denoted here. You've got your yellow restriction site, which is also denoted here. And you've got your ampicillin resistance or antibiotic resistance gene here. Again, you probably want to put the different base pair sizes. This helps you just kind of track it all in your head as you're manipulating this plasmid as to where the different pieces are and how big they are. And so I'm always a fan of put more information rather than less, because I think it helps, at least it helps me to not get confused when I'm talking about cloning and plasmid mapping. Finally, we're going to recircularize our plasmid again, which just means stick the beads back together into a circular pattern. And now we're going to cut with the green restriction enzyme. And so again, this is going to have the green restriction enzyme at either end, and then you're going to have your other pieces to label in your actual hand-drawn map. And that's what we see here. We've got our green restriction enzyme on either end of our linearized plasmid. We've got our yellow restriction site that's denoted here. We've got our pink restriction site that's denoted here. And then we have our genes. We've got our antibiotic resistance gene. Oh, wait, what's happening here? We've got our LAC-Z is actually split up from our linearized gene. So this is going to give you a great little key as to how this whole process works, because by cutting with green, we've now disrupted the LAC-Z. If you just recircularize the plasmid, your LAC-Z would also recircularize and be intact and would still produce a blue colony in any bacteria that took it up. But kind of keep that in the back of your mind as we continue to complete our pop bead cloning exercise. Now, we've talked a lot about the plasmid, and we haven't talked a lot about the chromosomal DNA, which is the straight, not circular piece, but the linear piece of DNA. And we want to talk about how many genes there are, how many restriction sites. And in this case, as per designated, in some of the write-up, we're kind of simulating how some of that Roundup Ready cloning occurred. And so we're saying that the red beads here are actually representing a mutated gene called ARO-A, which is involved in making those plants Roundup Ready. So in this case, well, let's see. We've got a pink restriction site, we've got a green restriction site, we've got yellow and green. So it looks like there's 5 restriction sites overall that are kind of a mix between pink, yellow, and green, and that we have one gene that we can see. So just like a plasmid, we can make a map for this. And since some of this information was provided to you in the write-up, when we make the map, we want to include as much of those little landmark pieces as we can. So, we've got our pink, we've got our green, we've got our yellow, we've got our arrow gene, and in the middle of the arrow gene is where one of those yellow restriction sites is. We've got another restriction site over here that's yellow, and then another one that's green. Because we have some of this information here, we want to go ahead and put anything that we have on the map so that we can kind of keep track of everything as we move forward. So now the POP Bead lab is asking you to look at the models of the plasmid, PPOP, and the genomic DNA that we're trying to clone. We're trying to clone that ARO-A gene, that mutated ARO-A gene. Is there one particular restriction site that's going to work for both? We want something that's going to cut the plasmid in a single individual location, not multiple locations, and something that will cut the genomic DNA in a way that it will remove the intact ARO-A gene. And so when you look at this map, obviously if we cut with yellow, we're going to be chunking up that gene, and that's going to have very limited usefulness. So yellow is definitely out. If we cut with pink, well, pink is only cutting one place far away from the ARO-A gene. And so really, as we kind of evaluate this and look at the potential options here, the only one that's going to work well is going to be cutting with green. If we cut with the restriction green enzyme, it's going to allow us to excise this genomic piece of DNA that will contain an intact ARO-A gene. And so if you cut with green, you then want to think about what size fragment you freed from that genomic sample. And so if we cut with green, it's going to be basically from here to here. And so it's just a matter of, again, just like with the plasmid, we're just going to add these up. And so 700 plus 600 plus 700 is going to be about 2,000 base pairs. That's going to conclude part 2 of the POP Bead cloning videos, and I will see you in part 3.